One.
Hi there.
Hello! Welcome to Microbiology Journal Club, where you nerd out big about all things
My name is
small.
Danny. In a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based at NYC dedicated to the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology, and I've mostly worked on bacteria, but I've also worked as a research integrity specialist, and I'm currently working as an editor for a scientific
Every
journal.
week, we meet to talk about microbiology, and today we're doing an overview of some of the coolest microbiology papers that we've seen this week.
And at the end of the month, we'll be selecting a paper to go do a deep dive into, so be sure to message us at microtwjc if there's something that has caught your fancy.
You can follow along with any of the papers that we discuss in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use comments or tweet us at microtwjc. And boy, do we have a show for you. First up, we take a brief look at SARS-CoV-2 strain A30, which is an old strain, but one that people have become worried about recently. We also have a new vaccine platform based on MERS. We also take a look at the complement system and how the age and shape of viruses can make them susceptible to being destroyed. And since it's Halloween, all you kids should be munching on candy. And we have a spooky paper on periodontitis and the bacteria that cause it. Plus, we have megaviruses, yeast evolution, and a new RNA sensor that can be used to detect viruses. But first, we'll be talking about the spike. Let's pull this up quickly.
The spike protein of SARS-CoV-2 variant A30 is heavily mutated and evades vaccine-induced antibodies with high efficacy. So that already sounds like a terrible, like a scary title of some sort. But yeah, and so A30, you said it was a strain from before. I don't recall hearing about.
Yeah, it was. It seems like it because from what I've read, it tends to be like kind of an older strain. That hasn't been spotted very. So like I think only 5 cases have been reported worldwide. 3 in Angola, 1 in Sweden, 1 in the UK. And it's tends to be because I think the A designation means it might have been discovered quite early on. I think
earlier, yeah.
In maybe, I mean it's the thing is it was discovered quite maybe not that early on actually. No, it was detected in 2021, I think.
2021.
And it's likely to have originated in Tanzania, where we don't have much like healthcare surveillance out there. People aren't out there collecting coronaviruses in the same way that they are in more industrialized countries.
Right.
So what they did, they looked at the spike protein specifically, and they looked at like the virus. So they used an expression system, so where they got a spike protein on a different virus. I think in this case they used a rhabdovirus of some sort. And they checked whether it could be absorbed into cells, how well it infected cells, and also how well it evaded neutralization by antibodies elicited by vaccines or the immune system. And what they found was that it's been hitting some news like headlines because it seems to be like more resistant to vaccines and other sorts of immune things than you would expect.
Yeah. So there's only one figure in this paper.
Yeah, this is a very short paper.
But yeah, in that figure E, I think that's where we're seeing they're using like serum from different vaccinated individuals, and then they're seeing its neutralization ability on these different viral strains. So A30 is the one in yellow there, and
yeah. So, like, for comparison, the other strain that we tend to be quite frightened of is B151. That's the beta strain, also known as South African variant, which early on in the outbreak caused a lot of, like scary stories because that was also found to be very resistant to neutralization.
Right.
But that also hasn't necessarily panned out as people would have expected.
Yeah. We saw, right, I mean, everyone probably is aware that we saw the rise of the Delta variant over these variants, these immune escape variants, and it was thought like it's because like of the transmissibility of Delta. So like that was like the bigger factor. Yeah. But as conditions change, there could be different features of the virus that are more important for spread. I think that's maybe where the concern is coming from.
Yeah, I think the concern is a lot about like whether our vaccines will still work, but I am starting to feel like the vaccine evasion isn't necessarily as important as we might have thought it was in the first place, because that whole vaccine evasion, that kind of hits in at a more severe phase of the outbreak. So that will like so not outbreak, like
Hmm.
infection. So when you're in the deep tissues and you're fighting off the immune system, that's when it comes into play. But the symptoms that will affect most people and have the most effect on the outbreak progression is transmission between most people, because a lot
Hmm.
of people, quite I think most of the people will have like he'll have normal symptoms, will not get a severe disease. So that will play a much bigger factor in the spread as far.
Hmm.
So that's why I feel like this is my personal opinion, but I feel like the strains that have been more immune evasive haven't really taken on that kind of scariness because they haven't picked up the mutations that make them more transmissible yet.
Right. Yeah, I mean, I
Um,
think that that's sort of the big question, and maybe that's the piece of
yeah,
information that tempers the scariness of this headline is that there have been strains that are immune evasive, but we haven't seen them catch on in the population, and the reason is unknown, I guess, right? Like, there's no definite reason to point to, but just
yeah,
from that heuristic, we're like, well, maybe yes, this is probably a concern, but like we're unsure in what context it will be like a very relevant one.
yeah. And another thing that was quite neat about the strain is that it seems to be affecting like cells in a different way.
Mm,
Uh, so in certain cell lines, so they tested a bunch of cell lines to see which ones are more able to be affected, and they found that that TMP. So we used to TMPRSS2. That's the furin cleaver that we're most
yeah.
like concerned about because that cleaves SARS-CoV-2 and makes it more transmissible and more able to spread in the lungs
Yeah.
because that's where it's usually found. But this one seems to rely on cathepsin L, which is a different kind of protease that's found in different cell types. So there is this thought that A30 might have a different cell affect different cells.
Hmm.
So that's that's kind of interesting to me from a scientific perspective. But there is
Yeah.
there's this other thing that this strain might already be extinct.
Mm,
They've only been spotted 5 times. It hasn't been spotted for a while
right.
since then. So it's probably not something we should be too worried about.
Hmm.
But it's still I find it quite interesting in its own way.
Yeah.
But
I
yeah, this
mean,
is
I
quite
I feel
a short
like
paper.
what and this is sort of like in the in the post vaccine, right? Like as the pandemic becomes more controlled in the world, I mean, right, it's still like out there, but like as it's becoming more controlled, there's a lot of interesting stories I think that still are left to be told about, yeah, like why certain variants appeared in different areas and then disappeared, right? There's no like
Mm,
unifying story about those types of things. I think it's like these one-off papers, they give us some insight, and then you basically get a story of every individual variant that's out there, and like it probably has a unique sort of mix of factors to why it appeared. So like maybe when these ones spread, there was something to it, but like it
yeah.
also petered out.
I mean, that is quite an interesting natural
Yeah.
experiment where you've got viruses which all have the same like ancestor, and then they spread across the globe and they pick up different mutations in response to different
Right.
situations. So I know
Right.
that with the beta variant, that was linked very much to the HIV outbreak in convalescent patients in the development of that
Yeah.
immunity. That so in patients where they're more likely to get a severe disease, having ways to defend against severe disease will benefit the virus more potentially. That's the story so far.
Hmm.
But I mean, there are like a million stories with these viruses.
Yeah.
So uh,
Absolutely. Yeah, the other thing that I think is interesting, or like, yeah, in the same vein of thought there, it's like, um, yeah, like what are the types of mutations that might occur, like in now the vaccine populations, right? And then how will that
mm,
affect populations that aren't as vaccinated, right? I think that to me, that's one of the,
yeah,
like there's still this idea that we should be preventing spread as much as possible, even though in some places people have kind of thrown that out the window because they're like, but I'm not going to get severe disease. But like, I would like to see some effort made, and people are, right. They're still masking,
mm,
hopefully, in some places, right? Like, to like control that spread, just in case, right? Like there is something that happens that's being selected for in vaccine populations that is going to be worse, right? Or like will have different effects on countries that aren't as well vaccinated right now, and then global travel's back, and it's just going to cause like these little complications.
yeah. And also thinking about animal reservoirs, like so we talked last week about the
Mm,
mink, but there are so in areas where there is a lot more like diversity or more like kind of interaction between people and animal populations, you can see that some strains might actually be more affecting animals,
Right.
and we might see spillover, so things like
Yeah.
that.
And then Minimal wrote in the comments just like also we don't have this insight about like other tissues that are picking up, right? That are getting infected by SARS-CoV-2.
Yeah,
What's driving its ability to get into other tissues? Are there mutations that could happen that make it better at getting into other tissues? And if we saw that, then like that would be also something to understand, like because we're trying to understand evolution, which is like super
yeah,
evolution as it happens in real time. Like that's like an incredibly difficult thing to do, right? Like people need a lot of like you need time to like and you need the resources to gather that data to start understanding these things. But again, like all of that data would add to our understanding and again maybe hopefully predict. I think I almost wonder what are the takeaways from like the viral surveillance people from this
hmm.
pandemic? What have they learned? Maybe that's like something that I don't know, we can keep our ear to the ground about. But like I assume that there might be signs, right, that people look for to be like, oh, this is
Hmm.
important for this reason or the other. I'm not sure, but yeah, that's something that I think is will continue to be
Yeah,
of importance as like even after this pandemic is well controlled, right? There's only going to be another emergent, right? Like the way that I guess our globalized world has been set up is that there's only and climate change and so forth, right? Like
hmm.
urbanization, people moving into places, right? Wild, so-called more wild places. Like there's just more chance, right, of spillover and these pandemic events happening. So understanding like
Yeah.
how to surveil for those things is important. And data like this is definitely like a little piece of that story.
Yeah, I mean, and one thing the thoughts I was having when I read this paper is like potentially, because it doesn't affect the lungs so much, it might be affecting different organs, and that might be a transmission cycle that can only be, but it's quite difficult because when a virus affects the lungs, it's a lot more visible, like it's a lot more dangerous to someone than if it might affect the gut. So it might be that they aren't getting sampled because of that reason potentially. So there's lots of thoughts provoked by this paper, but yeah,
What Minimal wrote like consensus sequence at a single time point turned out to be uninformative for this virus. Do you understand what he means by
hmm.
consensus sequence? Consensus
So I
sequencing?
believe that that's uh okay, consensus
Like
sequencing.
just like that they only
So
got the most prevalent virus from an individual.
yeah, and yeah, I think minimal does point out because it is only 5 time points, so they don't necessarily have everything solid. I think, um, uh, so, uh,
Oh,
and
you think it's
also I
about
guess
this
what
paper?
I think it might be,
Yeah.
uh, because I think like because I think we found that there is some change in SARS-CoV-2 like genome throughout an infection,
Yes.
maybe at least in
Yeah, there has
convalescent
been. Yeah.
syndrome.
That's what I'm
Yeah.
thinking. Like it's like, oh, just because like you only see what the virus is like in this one time point, you don't under like yeah, there's variance, but choosing the most common genome.
Yeah.
Yeah. No, that I mean what I'll say to that minimal is like that's true for a lot of viral infections, especially RNA virus
Hmm.
infections. And that's like that's an understanding that's I don't think it's being integrated clinically because
Hmm.
like you need a lot of data to say like oh here are all the different viral types within an individual but yeah there that it's an understood concept at least that that type of variation does lead to changes in the way that the pathogenesis of the virus yeah
Hmm.
no, yeah, so that you're right. Or Fuzz, we are thinking on that same line. Consensus sequence in this
Okay.
case is
Right.
talking
This.
just about that, like, yeah, a single virus from the sequencing. And yeah, that's true. It's true, right? Like in
Yeah.
HIV, I think to me, I first heard this story in HIV. They called them quasi species or something, right? I think
Right.
that's the term that they use in that field. And it's because that they're because of the nature of RNA and like the amount of virus, right? The amount of viral particles that are being made, there are variants inside an individual, and that could also lead to certain effects. But when people do
Oh.
surveillance, they
Yeah.
don't usually capture that information at that depth.
I mean, for HIV, it's really important because of it, because it evolves like resistance to antivirals as you treat.
Yeah.
So early on, I think there was like a very antiviral highly active antiretroviral treatments where they'd switch out retroviruses with different like drugs to treat the virus as it became resistant.
Yes.
So to switch
Yeah.
to and ramp things up until
Yes. So
eventually
there you
like
can see it a lot more
yeah.
clearly. And again, because there's something about that virus that is like more flexible in the evolutionary sense. I don't think like it's been observed in coronavirus, that those types of things, but they have observed the variants within an individual, and it's shown that there's like a bottleneck, right, in transmission. Like, but the relevance of that isn't known. Yeah, or at least we
Yeah.
don't know it. we
Yeah,
haven't
I
heard
think that we've
we
had
haven't
quite
heard the explanation.
yeah.
Yeah.
Yeah, I mean, there really isn't that much to this paper,
No,
but
no,
it's
it's just
mostly
like
like
a jumping off point for a bunch of other discussions.
yeah, yeah. It's another like fear-based headline that has been going around a lot, and so I just wanted to address that
Yeah.
a bit. But yeah, next up is Middle East Respiratory Syndrome coronavirus vaccine based on a propagation-defective RNA replicon elicited sterilizing immunity in mice.
Yeah, so we talked about replicons last week, and I was like, "Oh, cool! Like, it's a
Yeah.
nice idea." And I don't think we said like one of the applications of replicons is vaccines, because if you have like a single cycle of replication in cells, right? That would presumably activate all of the intracellular signaling pathways, right? That a normal viral infection would activate. And but then it wouldn't produce the next round of viruses to go on and infect more cells. And so that event, that like one replication event, could actually be an immunogenic event. And so in this paper that I found, they use replicons for MERS in a mouse model and show that it can produce some immunity.
Yeah, this is interesting. So they knock out a few like major genes, like the envelope gene, and I think some open reading frames. So like envelope gene is quite important for like building up the virus. So like I imagine that they end up with like wibbly wobbly viruses rather than like, but yeah, it's quite interesting because I'm just looking because I saw the mouse data. It's quite interesting because usually like they show like weight loss data and the vaccine will like kind of still produce a dip, but this one I think you don't really see much of a dip at all. So it seems like it does like not only it just erases all the pathogenic signs you'd expect from the infection, which is quite a positive outcome.
Yeah, absolutely. Yeah, I mean, I think it's if you if we're thinking about like what theoretically, right? Because again, we don't have the mechanistic information about this, but like when you think theoretically about what immunogenic signals are being seen, right, with different types of vaccines, like this type of vaccination strategy, like you're going to get the production of the spike, but you're also going to get the production of like those polymerases and those subgenomic RNAs, and like these are other signals that might help to create some immunity. So it's promising. Again, it's just in a mouse model, so like it's quite early in the R&D process, but it has that potential. And I think there were replicon-based. Now that I'm thinking about it, I believe I did see that there are replicon-based SARS-CoV-2 vaccines in the pipeline. Like I don't know how far they're going to get through the pipeline now, but yeah.
But yeah, I think the selling point is the idea that this is a proof of concept that can be applied to other viruses that are similar to coronaviruses. So that this strategy could be used for SARS-CoV-2, but also MERS.
Maybe any other
blanking on other
emergent.
coronaviruses.
Other emergent virus. I mean, I think in some ways it's also encouraging for maybe other viral. Like I know that you probably want to stick towards like the specific family of viruses or something to make some conclusion of. But I'm sure it also excites people to see this data and think about other viruses, right, that might have replicon-based vaccines in store for it. Or other Sorry, go ahead.
I think the cool thing about like coronaviruses, because they're proofreading, they can have much larger genomes.
Mm-hmm.
So theoretically, you could, if you can keep the replication apparatus, you can insert like other proteins of other viruses or other things in there potentially, because you've got more space
Oh,
to add
interesting.
in.
Yeah, so the replicon itself is a platform that has space for some
Yeah,
other antigenic objects. Yeah, I think that sounds like an interesting thing. This is like,
I mean, I don't know how that would go down,
yeah,
but
yeah. Well, I mean, like we do see that. I mean, I haven't picked up these papers necessarily, but I've seen a bunch of papers come out during this era of like everyone wants to publish about SARS-CoV-2, just about like all these different types of vaccine platforms, right? There's like a Newcastle virus-based vaccine platform.
yeah,
There's like other viral-based vaccine platforms, and a replicon, a beta coronavirus replicon could be one of those platforms, actually. Yeah.
yeah. Anyway, next one is in situ intraepithelial localizations of opportunistic pathogens Porphyromonas, gingivalis, and Filifactor, alocis in human gingiva.
Yeah, I picked this one out. It's an older paper. It was published a while ago, but I saw it on Twitter, and then I was like, oh, actually, I really like these. I really like papers where you get some human samples and you look at how closely the bacteria are associated with them in like from some sort of biopsy. And that's what they did in this paper. They have some, well, in part they did that in this paper. They have some human gingival samples. So like that's your gums, right? The space around your teeth.
Yeah.
And that's an epithelial layer, just like our skin, basically. I mean, it's a bit different because it's always wet. But yeah, supposedly it's supposed to keep stuff out. Like bacteria shouldn't get like 100% through it. But I mean, even in the healthy tissue samples, they stain with this some antibodies, and it's colored green in these pictures. I think you were just at in figure one. In figure one, yeah.
Oh, figure
in figure
one.
one,
Okay,
they have
figure one.
they have the healthy tissue. And like even in the healthy tissue, like there's quite a bit of this bacteria like scattered across the epithelial layer. There's a lot less that gets past the lamina propria.
Lamina propria,
Which is really the bottom of that epithelial layer. There's like some tighter junctions and things where not a lot gets through there. But then in the diseased ones, you can see there's a lot more that gets through the lamina propria. And then in general, like I think they see like just a rise in the amount of fluorescence that they're getting from this bacteria in general. So they have a lot more association and disease. And then they go on, they do some confocal, they do co-localization to look at it with other ones.
Yeah.
What figure was that? I didn't.
I think we're on figure 3 now.
So they zoom in and they
is...
also look at its co-localization with this other bacteria. And so there's this, and they see this co-localization particularly in disease states. So in some ways it's like a biomarker, maybe, right? Like you can think of it as like a biomarker that they're trying to identify because they have this, because they're able to correlate it back to human samples. And then they also do it I think they do like some primary cell culture of these gingival cells, and they show, like this co-localization between these two bacteria, we see it more often in a disease state.
Yeah. So P. gingivalis is around, like we've mentioned before, that's around the gums, but F. alocis tends to be like very hard to find unless you have the infection. And there almost seems to be a hint there that these bacteria are like cooperating in some way.
Yeah. I mean, like I think there's so there's this model in tooth biofilm, and I think it's like broadly applicable into other biofilms as well. But there's a succession of colonization that happens on the tooth, on the enamel surface, but then even in this like epithelial surface where it's like you have like the regular bacteria that you always have in your mouth, and they're like doing their thing, but then they make products or you eat things that they digest into other like products, and those products then produce the next surface that another bacteria can attach to. So you get this succession of things that are growing, and I think that they're trying to point within that model with this data, they're trying to point to the idea that yeah, like if you have there is a time point that's probably predicated by that gingiv gingivitis, gingiva,
Gingivalis.
gingivalis. There's probably a time when that like that's an early step, right, in setting
Hmm.
up these things, and then later on it gets right. There's growth of the F alocis, right, because it's attaching to something later that's being produced. I mean, they don't say it explicitly, right, but that's sort of the way that I might integrate some of this data into that model of like succession.
Yeah, so like it starts off where you're chewing your nice Halloween candy, which I imagine is candy corn
Yeah.
or other and then a bit of sugar gets stuck to your gums, and then bacteria will grow. And then you'll get a succession. It'll create an environment that's better for other
Yeah.
bacteria, and so on and
Yeah,
steps.
exactly. And
So
then
yeah,
those bacteria that are now growing in your gums, like those ones are also making like some acids or like maybe causing some inflammatory reactions. So like now you're going to see some pathogenic process start emerging. Yeah, and so that's
yeah,
the thought. But here, I mean, this paper doesn't give us any of those real conclusions, right? Really, it's just showing
right.
us like this instant, right? This snapshot of bacteria co-localizing on the human epithelium in the mouth. Yeah. And then they also do some effort to
Yes.
just say like these aren't dead cells. These are
Mm.
thriving living cells that are there, right? It's not just like, yeah, dead stuff comes in our mouth all the time and deposits and gets killed off. No, like these cells are growing in these places.
Yeah, so that's a take home message for the kids, I guess. If you want to have a zoo in your mouth with all these different wonderful strains of bacteria that want to decay your teeth, go ahead, go wild with gum.
Yeah, I mean that's why we brush our teeth, right? We sort of reset this process every time we brush, and like, yeah.
Yeah.
What was happening? Like, I was just thinking about it recently. I don't know why. Maybe I was looking at my teeth or whatever. But like tartar buildup, I think is so fascinating to me, right? Like sort of the harder mineralized biofilms that form in our teeth. Like I always wonder, like what, like we break them up as part of our dental hygiene now. But like,
Hmm.
I don't know. Like, is it really, is it that bad? Like maybe there's like nice good tartar and bad tartar. I don't know. That's just like
I
one of the
mean,
thoughts.
I I think that it's to do with the change in our diets because I think back in like Paleolithic times, we we're eating lots of hard nuts like that aren't very didn't have as much sugar as we do today.
Mm,
So there'd be natural wearing down of teeth,
yeah.
and that would be more of a threat
Right.
than like
Right.
tartar or
Yeah, we kind of like
gum disease.
uncovered this, right? By like removing a bigger factor that would impact, right, the design because like our teeth were evolved for a specific design, right? But our diet is actually a hugely different environment. And yeah, I can see that being a way to think about it. I like it. I like that.
Yeah. Next paper. Ooh, okay. Morphological transformation in respiratory syncytial virus leads to enhanced complement deposition. Um,
Mm-hmm.
So we've talked about a complement before, I think a bit. I think last time I talked about it, I described it as angry proteins that live in your blood.
Yes.
But um, yeah, so there's like a bunch of proteins that live in your blood, and antibodies can direct them against pathogens. And
Hmm.
so if an antibody binds something, there's a bit that can bind onto complement, and then there's like a whole different like let's say reaction. All these little proteins in your blood start talking to each other, and they can decide make a decision to like attack the surface of a bacteria and create a whole bacteria or a virus actually. So in this
Yeah.
case, they're looking at a virus, a respiratory
Yeah.
syncytial virus. Um, and this is a fascinating little paper because it looks at like how complement binds directly to proteins, how it's recruited by antibodies to the surface of this virus. So this virus has
Yeah.
two main important surface proteins. There's the F protein, which is the fusion protein, and G protein, which is the binding protein. So
Mm.
to bring this back to SARS, SARS has those both like in the same protein spike, both acts as a fusion peptide and as like the
Yeah.
initial binding. Um,
Hmm. Although we also, and it also might bind, yeah, and like in this way, it's almost like more a specific like receptor co-receptor style.
yeah,
Yeah.
and so what they did was they looked at antibodies. They looked at like the angle at which these antibodies can bind and try to work out like which antibodies are more efficient at recruiting complement based on what angle they did. And what they
Mm.
kind of found was that if the antibodies pointed more away, like from the surface of bacteria, this better at recruiting complement. And
Yeah,
then they
and
found some, yeah.
oh no, I was just saying, and then they integrate that understanding into this interesting thought, like the shape of the viral particle matters in terms of like what antibodies would bind better to it. Um,
Yeah,
mhm.
the, they found like because the viruses the respiratory syncytial virus has like a number of conformations. So when it first emerges from the cell, it has this rod shape, and when
Hmm.
it when it is left for a while, it kind of ages and then forms this like ball shape. So I think that's like one of the new findings they found in the study where because what they started off with, they found that there's a mixture of both, and they said, okay, well these seem to be affected by complement slightly differently, but why is that?
Hmm.
Why is it the ball-shaped bacteria viruses? Why are those being destroyed by complement more readily than the rod-shaped viruses? So then there's
Right.
a couple of bits of the paper where they rule out certain other factors that could be. So like, okay, is it because so one of the things they look at is the F protein. So that can change to its fusion conformation, or so it's got its pre-fusion conformation and post-fusion conformation that can be activated. I think if they mix around with each other a lot, and they found like with
Hmm.
this with the straight ones, it was just in the pre-fusion conformation,
Yep.
and so it's all fresh and new. And then they found the round ones that they had both pre-fusion and post-fusion. So they thought, okay, maybe that's why. They found out, nope, that's not the reason why. But they looked out and go, okay, why is it suddenly going? Why is this? There's this change in diversity, and they found out that actually, if you leave the rod viruses around for a long time, the actual like kind of structure of the virus breaks down a little. So
Hmm.
you've got like this what is it matrix protein that underlies the membrane, and that
Yep.
becomes dissociated from it. So that matrix protein gives it its rod shape, and when it ages, that kind of becomes a bit degraded, and it pulls away, and then like everything wants to become a bubble, so the virus becomes a bubble, and then they go, okay, what if it's just the shape? So
Yeah.
how do they do that? Well, they inflated the viruses. They added loads of like water to them. Basically, that's
Hmm.
they created an environment that caused them to take up lots of water to form into these balls before they had a chance to degrade. And then
Yep.
they measured it, and they from that they found that actually it was seemed to be the shape itself that was the problem. Um,
Yeah. And that's related, right, in that idea that like we're saying that these different binding angles, right, somehow
yeah,
affect, right, its ability to activate complement. Because if the shape of the viral particle is changing, it means that the orientation, right, the relative orientations of those fusion proteins are changing with each other, and that means like these sort of um, multimeric antibody molecules are going to have more difficulty grabbing onto them. Yeah, grabbing onto them.
yeah, they describe it as like a loss of curvature. So the rod is very tightly curved because it's like very streamlined, and then when it becomes a sphere, that curvature gets lost because it's a much more energetically favorable position. So,
Hmm.
and then the antibodies can actually get in between the F proteins much better, and the complement can fit in
Yeah,
and
yeah.
start attacking.
I really
So
like that because it's an interesting thought. Like I've always wanted, like why do viruses have these specific shapes? Why does anything have
yeah,
the shape that it has? And like this is a nice story to say like yeah, the shapes actually they matter, right? Steric, we talk about a lot about in chemistry, but it matters a lot here in the micro world, right? That yeah, steric hindrance of these molecules, like it's meaningful. These small things, like they do affect binding dynamics and thus the outcome of your immune system or a productive infection.
so yeah, this is what happens when viruses get out of shape. They get
Yeah, by taking on too much water. I guess they're
yeah, they
getting kind of
take
old. Yeah,
yeah.
their proteins are just sort of not as sticky as they used to be.
I've never felt so much more sympathy for a virus than I do now. Oh my gosh, this is describing me. I'm getting old. I'm losing my shape, and antibodies just randomly stick to me. It's just
One thing I also want to bring out in this paper that stuck out to me is that they use such a funny way of labeling these viruses, and it's a way that comes from from microbiology, sort of biochemistry, envelope biochemistry. And so we mentioned before that it's hard to make glowing viruses because
yeah.
like they don't have a lot of space in their genome to make a glowing protein for us. So in this particular experimental setup, the way that they get these viruses to glow so they can observe these shapes and so forth is they use an enzyme called sortase, which is used in gram-positives to anchor different proteins to peptidoglycan, which is like one of the major envelopes. And yeah, they use that to link fluorogenic substrates to the F protein.
Yeah, that's like really old school, and I can understand why they did that, because it doesn't put any like excess like kind of burden on the viral genome or whatever host genome.
Yeah.
So
Yeah, exactly. It's purely just like a little tag that they're able to add exogenously. And they do that in like the cells that they're producing these viruses in. So those cells produce the sortase and so forth. Yeah, I just thought that was really cool.
yeah. Okay, next one is Listeria monocytogenes. Sensitivity to antimicrobial treatments depends on cell origin.
Yeah, so this is also a pretty short paper, and it speaks to this idea that, well, first of all, Listeria monocytogenes, like people, you may have heard of this. It's part of the reason why they always advise pregnant women not to eat too many cold cuts, because it's a bacteria that it can cross the placenta. It actually crosses cell. It's able to sort of it swims, I guess it's propelled. It's propelled by your cytoskeleton through between cells. Like it actually pokes out from one cell into another. And it's well known to be this sort of like grows in cold temperatures. So specifically in these types of meat processing plants where everything's like chilled and like presumably it's cold, so nothing's growing, but like Listeria can like still persist on some surfaces, and then you like eat those cold cuts raw. And so like that's the method that, you know, people don't want Listeria to have outbreaks in these food packaging plants. And one of the things that they point out in this paper, and again, it's known before, is that the exact metabolic state, I guess, of Listeria in these different conditions can make it more resistant to some of the ways we might try to remove it from the environments that we're trying to clean up. And so specifically, they focus on this idea of like a small cell variants, or different ways that they can be grown, right? Different metabolic environments. I think they also temperature shock it at some point. So like these are different just ways of like, ways of growing up Listeria.
So yeah, can it survive a sous vide is the kind of
a sous vide and then being washed, right, with some sort of antibiotic solution. Yeah.
And I think the main thing is like a lot of these like measurements. So this is very interesting for people who are working in food standards, who want to make sure that food is safe. You need to be able to understand. Okay, how is Listeria? Is Listeria going to be able to survive? What's a safe temperature? What's a safe treatment? But different labs use different ways to store Listeria. Some can be stored on stock. Some can be left in a refrigerator, and sometimes it can be like in a. So I think it's almost a plea for transparency in that case, because what this finds that there is like a definite difference. So if you like pluck it from a colony that you've just grown overnight, compared to if you go and put it straight off of like a freezer stock, that's gonna make it different. So that means when you're studying one type of intervention. So like the intervention they're studying here is like a type of wash with some antimicrobial compound and the heat treatment. And based on the way that they're growing up their test bacteria, you get really different results. So it really like calls that we need like a common protocol for assessing the antimicrobial sensibility and even heat sensitivity of this bacteria, because otherwise you might like falsely believe that a certain method is safe when actually it just is. Without testing on all these different strategies, you don't necessarily know for sure if it's safe in all situations.
Right. And I guess that's definitely, I think, the main takeaway that this paper is trying to push forward. And then I'll also add in there is that this variance in the way that Listeria responds to different treatments, like you also have to think about like what is the type of right, like you'll probably see Listeria in different metabolic states all around, right, as a contaminant. So like there's that, it's actually related to a very real concern, right, inside of the plant as well, not just like from the test perspective, but that like when you go into the real world, you're not going to have like a consistent, way that bacteria was placed as a contaminant. And so, you have to make sure that you're testing a wide range maybe of different metabolic states for the bacteria. Mm-hmm.
Yeah, we do have to like figure out which one is most relevant and whether we go with the most relevant or the one that's like the most scary. So, for instance, do you want to like make sure you get in a situation where the Listeria is most likely to survive, and then if you can like kill the Listeria that's most likely to survive, then you should be able to kill off all the other ones? Or do you target against the one that's most likely?
Yeah.
So to
and
appear.
This is, I think in food safety processes, there are many, right? Like I think there's a concept of like control steps and things like this, right? Like everything can be dirty up until this point, and like this can sort of inform the way that those types of processes are put in place, and to also understand that like if all these different steps are being done in a non-clean fashion, then what are the types of bacteria that could make their way to that step, right? Because that type of bacteria is going to, like, that's the state that we want to test our cleaning step with, something like that. Yeah. Okay.
Right. Next paper is Phenotypic and Molecular Evolution Across 10,000 Generations of in Laboratory Budding Yeast Populations.
Yeah, so this is almost like this is the yeast version of like the long-term evolution experiment. So they have yeast and they passage it over and over again, and at set points they freeze down different points, and they do sequencing to figure out like what genes are there, what genes are gone, what genes have picked up mutations, what's happening to these yeast as we passage them. So they have 3 different environments that they do the passaging in, because as you would expect, the environment that you grow a microbe in over and over again is going to should have some impact, right, on its genome. So they have like they have one regular media condition at 30 degrees Celsius, and they have 2 other media conditions, one at that low temperature and one at a slightly elevated, like a heat stress temperature, I guess you could say.
So yeah, this is interesting because yeast is a eukaryotic organism, so it's more closely related to us than to bacteria. And I think the idea here is like with a lot of our evolutionary studies have been doing with that really long E. coli study, but E. coli
they're not like us.
they have they're not like us. Whereas yeast, they have a diploid genome. They've got I think 300 different genders or something like that. They've got a lot of they've got a lot of variation.
You said genders. It's like that means they identify, but like sexes.
Oh god, no, sorry. Yeah, I apologize. It's sex, yeast, sex. They've got 300 different sexes. Gender's a completely different term. It's got a lot of different other connotations. But no, yeah. Well, but yeast is the argument I use against like people who say there's just 2 because well, tell that to the yeast. Look at yeast, look at turtles, look at all sorts of other organisms. But that's off the point. What's interesting about the point is that how this is going to be modeling a much more different evolutionary, because I think deep diploids tend to be thought of as more stable.
Yeah, I think the problem is like you have that second copy because it's like a backup.
So we need to figure out like what exactly will happen, because will it be quite different, and how that might be relevant to other evolutionary models.
Absolutely. So I mean that's the impetus, right, to go down this path. And the sort of the experimental design has been set up by doing those early prokaryotic experiments, and then of course deep reading into the yeast literature to try to say like, yeah, like we've heard about this phenomena before, and we're seeing it emerge in this particular experimental system. And so they do end up talking about a few different. I mean, they talk about a whole bunch of different things that they observe. But the ones that stuck out to me was the diploid one, right? Where it's like, because they have 2 copies, there is this push towards becoming heterozygous for everything and accumulating mutations on one side, but not on the other. So like, I guess when you're put in this like set box for enough time, there's like a push for all this different variation.
Yeah, I mean I mean this is going to be quite interesting because there's this idea of trying to predict
Uh-huh.
evolution and how certain things can be predicted in bacteria, but the question is, do those same principles stay true for eukaryotic organisms? Well, we need to
Hmm.
test them, but that's going to take some time because it takes a lot more time for those sorts of things to evolve. So that's why they decide to do the 10,000 generations and keep it going for as long as they need to discover new
Right.
diversity in yeast.
Yeah, and they sort of pick up like a set of genes that they see where they see repeated mutations into them. So like that could speak to, I don't know, like the where yeast are like looking for flexibility, like where they have some flexibility. We talked about mutational hotspots last week, right? Like this could speak to specific hotspots maybe in yeast that like are
Hmm.
more right? Are set up for adaptation. Um, that's figure 6 is the what I was just talking about, multi-hit
Right. Let's
on
see
genes.
what figure. Okay, figure six. Right. That's right. Thank you.
Yeah.
Uh,
Yeah.
yeah.
Yeah, so like this is some information that you can come out of the system. They like in figure 7, they talk about a specific pathway that they find, and the like this idea like, oh, there could be like a certain more fit condition versus a less fit condition, and what's the trade-off, I guess, between those two things. And then I think what's the other story that I thought was interesting? Oh yeah, I said figure 8 is that idea that they're picking up a gene on a mutation on one side or in one copy, but not the other copy. Um,
Mm.
and then they even talk about these things. I don't even know what these are. The 2-micron plasmid and killer phenotype. It's kind of like they talk about specific, like I guess, curious genetic objects that exist inside yeast as not part of like maybe not the main chromosome, but extra chromosomal. So I didn't realize that yeast have just like
Mm.
plasmids essentially that stick around, and they're talking about like would these disappear, and that sort of makes sense, right? Like after all this evolution, like it goes away, but uh, it gives some insight into the question of like why do yeast have them in the first place if they get removed over this time? Like the pressure must exist somewhere, and it's just not in this long-term evolution experiment.
Yeah. Okay. So I think we can go on to the next paper, which is analysis of the first genetic engineering attribution
Yeah.
challenge.
So maybe this one's a bit more biotech than it is microbiology, but I just didn't even think that people were doing this. Like when I found this paper, I was like, oh, that's kind of interesting. So this idea that when people make genes, when they do genetic engineering, you're making these very specific choices of how do you bring
Mm.
together those pieces? What promoters are you using? What codon, right? What version of the codon do you use? And those choices that are made may actually reveal, like, may have some underlying pattern to them based on the person. It's based on your training, like what papers you've read, what you think is the best technology used at the time. So those things could be interpreted, and then tried to use and then used to assign authorship to mysterious genetically engineered objects. So I think there's a biosecurity slant to this type of research,
Hmm.
where the idea is that, can we make an algorithm, right? If we sequence something from the environment, or like, I don't know, some maybe a weapon or like something contaminant. Maybe this is also about like companies that are doing research on things, right? Like, and they accidentally contaminate somewhere, right? Like, we want to attribute blame. You could use this. You would potentially use some sort of algorithm to look at all those different choices that were made and assign it back to the original designer based on other things that they've made in the past. And so they have this. They set this up like a coding competition, right? Where they have a big training set of data, and then they have a subset of that set is held back. And the idea is that you try to train your algorithm on all these different, different genetic assemblies where the designer is known, and then can you correctly predict the one that existed? And so people have done this before, but there's like this. I guess the major problem is that you misclassify things over and over. There's a lot of misclassification. So the goal from this from this competition was to drive down that misclassification rate. On this particular computational problem.
Yeah, it's really interesting because the idea is that you hit everything like so because I'm just thinking of the B movie terrible movie plot where people go, there's a virus, who created this? Well, sir, I put it in my algorithm, it was this person at this lab or a student of that person.
And I mean, like, of course there's so many different technologies out there, like it's interesting to see that this is something people are working on. And I mean, if we're talking about SARS-CoV-2 directly, I think I found this from Twitter directly coming from this discussion, right? Of like, was SARS-CoV-2 made by somebody else? And like, they were trying to spin this paper in a way that it would help resolve that problem, right? Where, people are working on this element of things, right? To make sure that, that we can have this, I guess, intelligence on what's going on. But yeah, a fun coding problem.
I can see like a Templeton's Foundation scam in this way. Can you tell which genes were created by God or human, and then
because if you can't
give me money, Templeton, I'll do it.
If you can't attribute it to a designer, then what else could it be?
It's an interesting paper. It's an interesting concept because we do get like kind of gene hacking kind of hack, but we don't necessarily see the products of those in the published literature. So this is quite an interesting like way to go about the problem because it's kind of like part crowdsourcing and
it's a very popular the idea of incentivizing people on these types of machine learning, like this is definitely from like data science field way of working, right? Like that's like what's the famous example I know about is like Netflix, right? had a big prize for people who could improve their recommendation algorithm. and incentivize people to work on it. This is just like this way of working is definitely from the coding world. I love to see this like almost it's cross-disciplinary thought, right? About like taking that way of working and applying it into some emerging problems in biotech.
So the next paper is also kind of tangentially related to microbiology, but RNA-responsive elements for eukaryotic translational control. So this is it's a biosensor. So what the idea is that it's like an RNA molecule that is knotted up in a way that prevents a gene from being translated unless another RNA that it directly is meant to sense will bind to it. So the idea is if you want to sense, say, RNA from SARS-CoV-2, you put this molecule in, and when it encounters the RNA, it'll bind to it and then unravel and allow another gene to be transcribed. So this paper is
the IRES. So the technology that they're using is this IRES technology, or some modification of it. They're going to make them called e-toeholds. But that IRES tech comes from, I guess, to me, this is more microbiology related because it does come from like a very old like understanding of viruses, right? I think poliovirus or whatever has like specifically directs translation of its proteins using internal ribosome entry sites and sort of bypass some of the other ways in which eukaryotic cells activate translation.
So the original model was like based in bacteria, and they had to move it into eukaryotes. But of course, the eukaryotic system is a bit more complicated. But they found that other viruses like polio and I think Hep C, that an enterovirus, they use those as internal ribosome entry sites. So these are sites at the middle of it that can like grab onto the ribosome, and they use that as a way to kind of allow these proteins to be translated. And so I think it's a kind of an interesting technology. So in this, they use like some toy examples where they test it, see whether it can be good at sensing SARS-CoV-2 or Zika virus, and how sensitive it is to detecting those RNAs. And they show that it's relatively okay. It's quite good.
So specifically, like the IRES, the way that this tech works is like that IRES is disrupted by some piece of RNA. And I guess it could even be, the idea of them as a sensor is it could even be RNA that comes from, right, just like the regular replication of a virus. Am I understanding that correctly?
Yeah, it could be any virus, any bit of RNA on the virus could trigger it. And the potential of this technology is that they can actually potentially nest like multiple ones. so it could respond to maybe one or two different sensors.
Hmm.
So that's quite important for things like cancer or other sort of diseases where the cell starts like behaving differently and producing a different set of genes.
Yeah.
So having a sensor that pick up multiple different RNA transcripts could be able to sense like when a cell might need to be receive treatment and allow for like specificity of the kind of production of certain drugs, maybe.
Yeah. Yeah.
Um,
Yeah. And I guess like in some ways this is about engineering the iris, right? Like
yeah,
understanding like it's sort of a complicated you can see the picture of it. It's a rather complicated structure of RNA. Like it has all these stem loops and things. I think they call it a
yeah,
pseudoknot at one point. And
yeah.
understanding like what flexibility they have to like change a sequence in it to make these iReses sensitive to different mRNAs, and then
Hmm.
actually showing that it works, like engineering style.
Yeah, so it's the idea is that they want to get more specific. So this is like falling into the idea of smart drugs, where you want something that can target a disease very specifically and not have off-target
Hmm.
things. So in this case, it would be like targeting Zika virus or SARS-CoV-2 virus cells only, and then having no effect on other cells.
Right.
Um,
But you'd have to have this construct in every cell, right? It's just
yeah,
kind of weird.
so that's
I,
yeah, that's where it kind of falls apart because how do you deliver this construct into every
yeah,
cell?
I don't like that narrative of like its potential application.
But for something like a form of chemotherapy, that could be a very
Yeah,
like a really
I
good
can see that. Okay, yeah, yeah,
yeah. So you'd have to be in like a very severe position in order to want to go through like having a line of this stuff pumped into you.
right.
But cancer is one of those areas where, and they were talking about like using these as building blocks to build like a transistor or a
Yeah,
sensing
yeah. I
system
like that. I think
for.
to me, like, when we talked about the metabolic engineering of yeast, right? Like, I think that this,
Mm,
to me, this technology fits into that world much easier because, like,
yeah.
as I may have mentioned in that in last week's episode, like when you do this thing and you put a new pathway in and then you find oops, like there's a collision here, we're making too much of this one thing,
Mm,
it's creating a bottleneck that's killing off the cells. Well, now you can have this little switch that makes sure that like the step that actually makes that right is now controlled by something that is dependent, right, in a different pathway that like makes sure that that bottleneck doesn't exist. I'm sorry, I maybe didn't explain that properly, but like
yeah.
you're relating the production of your synthetic thing to maybe something that's being produced endogenously in the yeast metabolism, and then that makes sure that that bottleneck is being regulated in a specific fashion.
Yeah, and I think you've looked at things that have been acting like transistors, but this one seems like a lot more like
Very straightforward. Yeah, it
refined.
seems like
So
pretty, yeah, very
very
straightforward. Like
straightforward.
because all you have to do is like design this little section of the Ito hold to resemble something. I think one of the first things they use as the RNA for it is GFP, right? So like
Yeah.
like that's like a totally artificial RNA that floats around. You can imagine trying to engineer your Ito hold to be like sensitive to some, important mRNA that's being made in a certain metabolic process. So now your artificial system is under control of is in the control of a natural system, an endogenous system, something like that.
Yeah, so that's what got me kind of interested in this.
Yeah.
But yeah, I don't think I've got much more to say about this, so that might bring us to the last
Hmm.
paper, which is a new family of megaphages abundant in the marine environment.
Nice. I saw this paper
So
too when I was searching
yeah,
through. I'm glad you picked it out.
yeah, this is interesting. So megaphages are bacteriophages. They feed on bacteria, and we've found bits and bobs, so bits of their genomes strewn across like various different kind of
Mhm.
studies. So we know they exist, but we don't necessarily know what they infect or how they necessarily
Yeah.
work. This paper did an investigation in the English Channel, probably one of the least glamorous stretches of water in the world.
I
Um,
find it glamorous.
it's
It's
so
because you're so
unglamorous.
close to it. I'm
Yeah,
just like, ooh, English Channel, so much
it's
history, all those boats going across for war and so forth.
yeah.
Foreign trade.
And now we've got a tunnel, so we don't have to look at it anymore. We just
One man's trash is another one's treasure.
yeah, true. I imagine it must be really exotic to people who haven't lived in
Yeah, yeah.
England,
I'd be like,
haven't
what? I want to go
done
through the
the
English Channel tunnel. I
yeah.
don't want to take a ferry across.
Yeah, go take a ferry across and take all the like cheap alcohol from the Carrefour just outside of Calais and play the arcade, the like decades old arcade machines on the PLO boats if there's those boats are still around. But yeah, it's a it's a it's a once in a lifetime
But
experience.
regardless of what
But yeah,
the human culture that's surrounding the English Channel, there's this like diversity of bacterial or microbial life in there that's untapped.
yeah. So yeah, they sampled the English Channel and they found like a bunch of these. So they found some sequences that they put together into a megaphage. I think they brag this is like the first like high quality draft viral genome for like this megaphage, which they could then use as a framework to search into other data that's already been collected to see whether this family appears in other
Yeah,
studies.
just really big genome phages.
Yeah, just really big.
Which again, I guess if we're thinking
and I
in
think
the technology, if we're using that technology lens to look at this, we could be like, oh, like that means more like cargo capacity to put in the genes that we want potentially. Or if you take the lens of like, you know, we're finding new life, we're like, my gosh, what do they use all those genes for? What is their life cycle like that they need this much information?
yeah, that's like one of the big questions of this paper. So they take a bunch of those genes and they sequence and they try to predict what their functions are. And a lot of them are related to the carbohydrate metabolism. So the thought is that they might play a role in the carbon cycling of the ocean. When they infect something, they change its metabolism and the way it like absorbs nutrients. So there's like one gene that they think might be responsible for increasing carbohydrate uptake during an infection. So whatever it infects, it makes it more hungry.
But I mean, I think that this is so. I mean, I love this way of thinking about genes, and especially like when we think about genes that are inside of phages, right? There's this concept, right, of like accessory genomes, right, and the core genome that like bacteria, they're constantly being bombarded by different phages that inject different genes into them, right? Those phages have like an evolutionary relationship with those bacteria, and so those bacteria could have an evolutionary relationship with the genes inside of those phages.
Yeah, it's an interesting thing because you can learn. Cause I think we've looked at this before. You can learn about the bacteria that a phage infects from the phage. Like you get because especially if it's got a CRISPR-Cas system, which unfortunately this one doesn't. But if it did have one, we could like get little segments of the bacteria it infects.
And I think
in fact,
the fact that they were able to assemble a genome from like in this paper, and what they're saying is that now they have like a scaffold in a way that like they can be like, oh, like maybe if we see hits to genes that are similar to this, we can think about maybe they assemble into like a megaphage as well. Because definitely. I think to remember, like in this particular sequencing strategy, this is the shotgun style, right? Of just like sequencing like just bits of water and seeing what comes out there. And so like they don't know what piece of DNA goes to what cell, and the reason they're able to bring it all together here is just by those overlaps, I believe.
So I think that brings us to the end of the show. It's been really great.
Sorry, I just.
I mean, we've had a lot of adventures, but to everyone, have a happy Halloween.
Absolutely.
And
join us next week for more cool microbiology news.
And we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Sounds about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
You can reach out to us over Twitter at @microtwjc. We both believe that peer review is a process and that we can all participate in. So we hope you had a good time listening to us ramble on about microbiology today. If you think you have something to add or found something unclear, just let us know.
It's been a pleasure chatting with you, Danny. Tune in next week for more microbiology content.
Bye.